So today we're going to be doing this machine learning for transcriptomic data workshop that is intended for people to quickly get through the basic concepts of RNA-Seq and then continue on to explore the table of expression using machine learning methods, supervised and unsupervised methods, as well as being able to visualize the results and show specific issues of interest within the data set that we analyzed. First of all, to make this possible and to do this quickly, we are going to be using the MULTI-OMICS data analysis and processing platform called t-bioinfo. The platform was designed for processing, analysis, and integration of multi-omics data, and it is used in research, so this is a real research product. You will see that on the platform there are many different sections and many different data types. Today, we will focus on transcriptomics, so the section that will allow us to process raw RNA-Seq data, and also to establish some differences within the data, such as differential gene expression on quantified mapped reads. The second section that we'll be talking about today is data mining. And under data mining, you see that there are two subsections called supervised analysis and unsupervised analysis. And those two methods, those two types of methods, can help us first explore and visualize these large data sets, as well as then build classifiers that allow us to identify gene signatures responsible for division of this sample group of samples into groups. Finally, we'll also be using this utility section, and this utility sections allow us to annotate the gene names and then understand how the expression of these genes can be mapped onto pathways and help us interpret the biological meaning of what we identified. So let's go through this in a little bit more detail. First of all, processing raw reads and generating a table of expression. To do that, we will build a pipeline, and then we will take a look at how these outcomes, the outputs of the pipeline could be visualized and what can we see within that table of expression. The second part that we will focus on is unsupervised and supervised analysis of the gene expression table, where we'll look at things like principal component analysis, clustering, and then under supervised analysis, we will do differential gene expression analysis that will help us select specific genes, but also we'll look at classification methods to determine which group should a sample be associated with. Finally, we'll look at the annotation, where we can select a number of genes and convert them into gene IDs, so simple name of the gene, as well as gene ontology of that gene. And then we can use differential gene expression output and map it onto pathways, seeing which genes are upregulated and downregulated on which pathways. Before I continue, I'm just going to make sure that everybody's with us and that everybody can hear what we are doing. So if you have any questions, we will try and address those questions in specific times between the sections. Let's just make sure that everybody has everything technically working, so you know where the chat box is. You have the presentation. You also have the files that we're going to be using here. And we are also going to be putting this workshop in a video format at the end. So if you ever want to go back and review the material, you will be able to do that. Going back to our presentation. To illustrate many of these things, we will be using a single data set. And this data set is a publicly available data set around the topics of precision medicine. Why precision medicine? What are we going to be looking at, and how is this relevant to the type of data that we are analyzing? First of all, precision medicine in the context of oncology a lot of times is around the challenge of classifying a specific subtype of a disease. In our case, we're going to be dealing with breast cancer. So here you see a mammogram, and you see this white spot, and this white spot is a tumor. And so the challenge to understand what type of cancer this is, we can look at, first of all, the origin of the tumor. So what tissue is it coming from? And you can see in the mammogram that it's not so clear where it's coming from. But also we have other tools. For example, immunohistochemistry allows us to look at expressed proteins by staining the tissue. But the limitation of this is, of course, that you can only stain the tissue so many times, and so you're limited to the number of expressed proteins that you can look at. The next step in our ability to classify samples is around gene expression. So here you see a microarray expression of 50 genes, and you can see the high expression in green and the low expression in red, and you can see how they vary between different subtypes of cancer. Why is this important? First of all, it's important because we're looking at predictive signatures that allow us to differentiate between cancers that are more aggressive, less aggressive, and are related to the survival, but also at specific issues around recurrence. So cancer does not come once and then disappear. A lot of times it comes back. And so it's important to understand what is the likelihood of a specific cancer to come back. Here you can see an example of a survival curve and a recurrence curve for patients, and a comparison of two panels that allows us to do that. So the panels, each one contains a different number of genes, and based on a specific pattern of expression, we can classify patients into high risk, low risk, and medium risk. Another use for this, to understand what subtype of cancer we're looking at, is selection of therapy. We can use therapeutic interventions like surgery, radiotherapy, and chemotherapy. All of these have different effects on the cancer itself, but also on the surrounding tissues, and so we can use them more appropriately if we can classify the cancers in a more precise way. The other aspect of precision medicine or personalized medicine is to understand the biology that is unique to the patient, and we can then prescribe drugs that are targeting specific biology and intervening in a process. Now, this is a much more complex process where we need to understand the unique aspect of the cancer in the context of a unique patient. And so some of the things that we'll be looking at today are related to this concept as well. In fact, the paper that we are going to be using and the associated data set is a paper that is called Modeling Precision Treatment of Breast Cancer. As you can see, these are cell lines. So they are cells derived from a tumor from a patient, and then they're grown in a cell culture, and that cell culture represents what is happening in the tumor. But at the same time, these tumors are treated, or these cell lines, these cell cultures are treated with different types of therapeutics, so different types of molecules. And then we also have a data set that measures the reduction in its ability to proliferate. So the division of the cells is stopped by these different therapeutics, and we can compare them, how they interact with different cancers. So now we're not going to be looking at every aspect of this data set. We're not going to be using the multi-omics, and we're not really going to be looking at the treatment, but we are going to be looking at the different types of cancers represented in this data set in this paper. All of you should have these input files. So let's just take a moment and check that you do have them. It will be a zip file, and it was downloaded. If you downloaded, then you can see in your browser. Here, I'm going to open mine. It's called workshop_sn_May. And then inside you have multiple folders. So please let us know if you don't have the files. That's an important part of it. So the other part of it is you need to have access to the platform. So if you go to the platform, server.t-bio.info, and then you should have received login information. So you can go ahead and log in right now. And after you log in, you will see this table of areas of analysis. So if you don't see this, please let us know and we will help you get there. Okay, assuming that everybody has the files, let's go under Input Files and Expression Quantification, the folder called Expression Quantification. And then there you will see this file called samples.svl. Okay, so if you don't have the files, where can you get the files? You can go on this page. So I will put the link right here. And then if you scroll down under the video, you see this button called Download File. And in fact, it will just be this link. So if you download this link, you will be able to see a zipped file. So it will look something like this: workshop_sn_May. Then you open this file and you see input files right here. And then under input files, you will see expression quantification and samples.svl. Okay, so what is inside this file? Inside this file, we just simply have links to FASTQ files. So you see that it's .fq, that stands for FASTQ, and then you will notice _1_2. That means that it's pair end files. And so each file will be repeated twice with a different extension at the end. If you have any questions or you don't have this, again, let us know. We'll try and help you. Now, before we continue to the next section, I just want to point out that a lot of this material is also covered in our online courses. We have 3 courses dedicated to transcriptomics, and they go from easy or introductory, transcriptomics 1, to intermediate, transcriptomics 2, and to advanced, which is transcriptomics 3. These courses cover the basics of converting raw files to gene expression table, analyzing that expression table using standard approaches like student t-test, and then additional approaches used in differential gene expression, as well as a more advanced course on machine learning that introduces unsupervised and supervised approaches to interrogate that data and mine it, and then visualize the results. Okay, if we don't have any more questions, I'm going to continue to RNA-Seq and the processing of data. What we will try to do here is take the raw reads, the files that we have in the SVO file, and then create a pipeline that converts these raw reads into a table that we can actually look at. So before we do that, let's understand a little bit what are we dealing with. So this is the genome, some sequence in the genome. And we have in this sequence a number of genes. There's a gene A, gene B, and gene C. Now, not all genes are going to be expressed. So we will only have, for example, in transcript A and transcript C. Those, in turn, are turned into reads. And so we have many, many reads that represent many genes. So one of the challenges that we have is mapping those reads back to those genes and then quantifying the reads aligned with specific genes. But it's not so simple, in fact. There are some additional details that we need to consider. In fact, the real process starts from shattering the RNA. So we have these reads, and then we use adapters to match to those different reads. And then we use PCR to amplify the reads. So we have many, many reads that include the adapters. And there's some effective PCR amplification. And then finally, we have the process of reading. Because of this, we are going to look at three components of RNA-Seq data analysis. The first component is preprocessing. So we want to remove the adapters, and we do some additional trimming, and then we want to remove technical PCR duplicates. The second part is mapping. After we've removed the adapters and PCR duplicates, we're going to map the reads on known transcripts, transcripts that are associated with genes, and recorded in a GTF file. Then we're going to map on genome. That's an optional step. Specifically, it's useful for identification of novel transcripts, so a variation of the known genes. And then we can look at a combined strategy, a strategy where we will create a pipeline that will give an output of all the known genes as well as transcripts. And potentially we can even go as far as just a table of exons so that we can deal with unknown sequences that might be of interest. At the end, once those reads are aligned, we can quantify them. And one of the questions is going to be around how to quantify them. Do we just count the number of reads? Or do we account for certain differences in genes, for example, their length, that will have an impact on the variation that we want to study? So now this is the picture of the RNA-Seq section. And you can see that the same color coding, blue, red, and green, is going to be on the platform. You will see that the blue buttons are associated with pre-processing. The red are associated with mapping, bow tie 2T. And the green is associated with expression levels, so quantifying those reads. Now let's go to the platform and create our first pipeline. Let me know if everybody by now has access to the platform and has all the files. Okay, I'm just going to give a moment. If you want to ask a question, you don't have anything, just go ahead and ask right now. Okay, if not, let us go. So RNA-Seq, I click on it. A very similar interface is common to all of the other sections. But let's take a look over here. First of all, on top, we have the inputs that we're going to be using for the files that we load into the pipeline. So we have a selection here for our model genome GTF, which means that we're going to be using a model reference genome with an associated prepared GTF file that contains the mapping or the positions of all of the genes. If you are dealing with a non-model organism, you would, of course, choose reference genome GTF if it is annotated, or you can also use a non-annotated. The second thing we have here is single-end or pair-end reads. So in our case, I showed you we have the _1 and 2, therefore we have pair-end reads, so we're going to select PE. There are a number of different types of files that the platform can handle, and we're working with FASTQ files. And finally, we're going to be using the human genome GRCH38. Now, once we click Upload Files, you see that there are a few options. You can upload individual files. You can use reference links such as SVL, HTTP, and FTP. SVL is a reference file on the server that you are using. And we are going to use URL bulk upload because we already have this file with all of the links inside a single file. So I'm going to go back to our input files. Somebody here, I'm going to, okay, sorry, Manish, put you on mute. Okay, somebody else is calling in. So again, if you have any questions, uploaded. We will select define groups check. In our case, we're not going to be doing differential gene expression, so we have to say cancel no groups, and now we can build our pipeline. Okay, so it looks like Manu can't log in. Did you receive the login details? It would be great, Sahil, if you can try and work with Manu to get him on the platform.
Sure, yeah, I'll do that.
Okay, if anybody else is having a difficulty to log in, you should have received an email. And in this email, Sahil provided you with access to the platform. Let's see. So you will get a platform URL, and then you will have your username as your email and some password that he emailed to you. So again, if you don't have that, just reach out to Sahil and he will try and help you. In the meantime, we were able to upload the SVL file and start creating the pipeline. So I will start with clicking on Start, and here I can read a little bit about the process of RNA-Seq. And then I have this pre-processing section. So the blue section that we talked about, Trim-O-Matic is a method to trim technical sequences. So there is a database that stores these different sequences, and then the algorithm removes those sequences, and you can read more about this algorithm by clicking this link. After Trim-O-Matic, we are going to remove the PCR amplification. So it again uses a statistical approach to clean all duplicate reads from raw sequencing data. If you would like to read more about PCR amplification, you can go to this link. And so now we've connected these two buttons. And after we connected these two buttons, we have cleaned reads. And so the next step is to map those reads onto the known transcripts. So we're going to be using Bowtie2, and later on we'll look at an alternative to this pipeline. But if you want to read about Bowtie2, again, it's a fairly complex approach to identifying where the read should be mapped. You can read this publication and understand a little bit more of the algorithm behind this method. Click Save. And then after we've mapped them, we are going to be quantifying the reads, the alignment of reads, the number of reads that fall under a specific region in a type of a number. So we'll talk a little bit about what those numbers are. In the meantime, here's an algorithm that provides several options, and it's called RSEM, and you can read about this algorithm right here. Now, what are the options? We can either count in FPKM, expected count, or TPM. In our case, let's use FPKM, and then later on we will talk about what are the different approaches here and how do they relate to our ability to quantify gene level of gene expression. You can also see that we have the option to create an exon table. In our case, we don't need to do that because we actually are mapping just on the full known transcript. And so we can click End. And this is RNA-Seq pipeline. Of course, if you want to, you can not even do the pre-processing steps, but we will see a little bit later in the presentation that it does have an impact on the output. So let's call this, you can call it whatever you want, but I'm just going to call it RNA-Seq simple pipeline. And click on Save. Okay, seems like everybody is on. And let's run this pipeline. Now, running this pipeline is going to take a long time, so we're not going to wait for the pipeline to finish, but just to make sure that you understand on the platform, every time after you create the pipeline, you need to click Run Pipeline. And if you didn't, you can also see under My Pipelines, there are prepared pipelines that look like this, and then there are processing pipelines and completed pipelines. So if yours looks like this, that means you have not started it. If it's looking like this, that means you already started to process that data. Now, we're not going to wait for the pipeline to finish, so I'm going to show you the output of the pipeline, and then we can talk about what is inside the table. But let us return to the presentation for a second and do a little bit of practice. So before we look at the output and some of the considerations, please try and create a different pipeline. I'll do this with you, but if you want to try it on your own, go ahead. We will use the same SVL file. We will take some pre-processing steps, Trim-O-Matic, and then PCR clean. We will use a different mapper called HiSat2. We will then create some isoforms using cufflinks, merge them onto a GTF file using cuff-merge, map this on transcripts using bowtag2t, and then quantify the reads using RSEM-X table. Okay, let's do that. Again, I will go under RNA-Seq. If you remember the settings over here, I'm not going to explain them again because we just went through all of this. But remember that we're going to be using the same SVL, so it's URL bulk upload. Let's not do any groups, so cancel no groups. Start, Trim-O-Matic, PCR clean, HiSat2, cufflinks, cuff merge, bowtag2t, RSEM expression table, and then end. Okay, let me know if you successfully created a pipeline. If you have not, maybe you need a little bit more time to go over the pipeline, so let us know, but we will do a few more pipelines during this session, so you'll have plenty of time to practice. Okay, here's an example pipeline that you should have received by now, or you should see on your screen, right? So if I take a look over here, again, I can see what I see right here. So what did we do? We uploaded the same SVL files. We trimmed the technical sequences. We removed the duplicates from our PCR, mapped it using a different mapper. Then we combined those mapped reads into a GTF, and then mapped that onto known transcripts and received an expression table. Now, we also had the option to add the exon table here, but we're not really going to be looking at it in this session. So you can, but you don't have to create that for your pipeline. Now let's take a look at the output. So you can see that the output table is going to have IDs of genes and then sample names on top. And so the columns are associated with the samples, and for each gene, for each sample, I have a specific number. Okay, so let's go ahead and look under our downloaded files. Instead of input files, we will look at output files. And under expression quantification, you will see two files. So you will see FPKM and FPKM not filtered. Now these are TXT files, so they are tab delimited. But if you're on Mac, you can simply drag this in Excel. If you're using Windows, you just do a right click and select Excel, or if you have any other table manipulation software, that should also work. And you see the table that we just saw in the presentation, ID of a gene, and here's an example gene, and then we can see some numbers, and we will talk about what these numbers are here in a second. Okay, so as you can see, this would be a single sample. So how many samples do we have? Okay, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 samples. Okay, then we have genes. Okay, so let's look, how many genes do we have? Okay, so each one is a gene ID. how many do we have? If we go to the end, we will see that we have about 20,000 genes. So these 20,000 genes, and let's look at the numbers. Okay, so what is this number? So let's go back to the top, and let's take a look at the numbers that we have in here. Any number that you select. So what does this 10.12 really mean? Right, so in this example, you see 3.27, so it's right here. So what is this number? Okay. Let's take a look at the different methods we have to quantify the number of reads that were aligned to the gene. First of all, we have the option of just simply counting them. The second option is called FPKM. That's the one that we use. And that stands for fragments per kilobase per million mapped reads. In other words, we are aligning this to have a normalization for the length, right? So we are just taking 1,000,000 mapped reads as our baseline, and then we are using every count, adjusting it to what distribution of reads we have in general. And then a very similar approach is TPM. So instead of doing fragments, we can do full transcripts per million. And you can learn more about this if you go into the RSEM publication and read about the algorithm itself. Now, another thing to consider is whether we are using linear scale or log scale. A lot of times you will see that the initial output is going to be in linear scale, but it's very useful to normalize it by applying log scale to the numbers. But once we do that, we also need to understand that now it changes the proportions that we are looking in terms of the differences. We will reduce the numbers, but we will also therefore reduce the differences, and we just need to remember that. Okay, so let's do a quick review of what we did here. First of all, we looked at pre-processing steps, removing adapters, and PCR duplicates. We then went over different mapping options, known transcripts, mapping on genome, and potential identification of novel transcripts, and then a combined strategy. And then finally, we looked at quantification of expression levels. Now, the question about using the pre-processing steps. As you remember, we used at least 2 methods of trimming and reducing duplicates. Does that really have an effect on the outcome? So you can see here 2 graphics. The first one represents a level of expression on you see over here log scale, okay, no duplicates removal, and no pre-processing. So they are perfectly aligned, so there's no differences between them. Over here on the right side, we see that we have standard pre-processing and no pre-processing. So is there a difference between them? And yes, we can see a difference right here. So in this corner, you can see that if we were not doing any pre-processing, our understanding of highly expressed genes, values of highly expressed genes, is going to be skewed. And so that's why typically you would want to use pre-processing steps when you are trying to analyze the differences between samples, for example. Okay, now if you have any questions, let's go over them now. I'll just remind you that a lot of these things are described in detail in our free online courses. Transcriptomics 1 and 2 deal primarily with the issues that we have been discussing today. Let me know if you have any questions about what we did so far. So you can either speak, or if you want to type them out, That's also fine. Okay. No questions so far, it seems. Let me then go on. So here are some of the questions that we had in previous sessions. As you saw on the platform, there are a few other options. There's error correction. There are different mappers. And so the question would be, okay, so why do we have so many different mappers? And then we also have a differential expression section and segmentation, right? So we have differential expression and segmentation. By the way, before we continue, I just want to mention that you will see this tau sign on some of the buttons. The tau sign represents an algorithm that was developed at the Tauber Bioinformatics Research Center, and it's a custom approach to this general topic. Other algorithms, you can download them online. They are open source, and you can run them typically in a Linux environment. You can run them, and if you know how to give them the right input and the right modifications to the inputs, then you will get the same output. And what this platform does is it helps connect via visual interface all of these different methods into a single pipeline and run it on the servers that we support. And you have 14 days to use the platform for free. And if you are interested in using this platform for research or for education, you can just let us know, and we can help you figure that next step out. So error correction is typically a section used for microarray data and low-quality sequencing data. We have different mappers, but they are organized by their function. So you can click on a section and read about the section itself. And then the other issue is with the exon tables. We reviewed that. Differential gene expression, we will actually create a differential gene expression pipeline later on. And then segmentation is a section that is useful in specific cases, but as you can see, a lot of the algorithms are custom, so we're not going to be talking about them right now. Machine learning. So now we're going to review what to do with this expression table using machine learning. The person presenting this will be Stepan Neosisian from the Tauber Bioinformatics Research Center.
Hello. So let us start from understanding what is machine learning about. And generally, machine learning is separated into two ways. First is unsupervised machine learning, and second is supervised machine learning. And first, let us discuss unsupervised machine learning approaches and what it's all about. Suppose we have a lot of samples. In this example, our samples are some PET images. And actually, we don't know which PET is on which image. So we don't know that this is a dog, this is a cat, and this is a dog too. And the aim of unsupervised machine learning is to mark these samples. So for example, we can mark them into this way. So these are dogs and these are cats. So in unsupervised machine learning, we have unmarked samples and we need to mark them. For example, we can draw them on the plane. So you see that dogs are distinct from the cats. So we have group 1 and group 2. And sometimes we can have outliers, for example, cat dog, which can be in each of these groups. And in our case, our samples are cell lines and our features are genes. So we have gene expression table. And first method of unsupervised analysis is just to draw our samples on the plane. What's the problem? In our case, we have 7,000 genes, 7,000 features. So we can't draw them on the plane because our samples are actually points in 7,000 dimensional space, and it can't be drawn on the computer. So our aim is to have some projection of this data to the plane, but not all the projections from high dimensional data to the plane are actually good. For example, do you have a pen here? Okay, let's look at my finger, for example. Okay, or to the cup. Yeah. So this is a three-dimensional object, and you see actually two-dimensional projection on your screen. So for example, I can turn it this way, and you see just some circle. And you can understand that this is actually a cup. But if I will turn it in such way, you see the optimal projection, and you can understand that this is actually a cup. So the aim of principal component analysis is to perform projection of high-dimensional data to the plane in order to maximize information which is associated with the data on the plane. So we can open our platform and try to run principal component analysis on the expression table. Here we have section data mining and unsupervised analysis. Then we need to upload files. Here our files are not so big, so we can do